All right, let's get started. Just to verify, can everyone hear me and see my screen? Right. All right, perfect. I'm gonna go ahead and get started then. So today we are covering cell division, and my name is Nikhil, and we are doing this through MCAT 101. So please feel free to email MCAT 101 or me if you have any questions in general or about today's presentation. And also, if you're not on the newsletter, please send me your email and I will get you added to the list. But before we talk about cell division today, I do want to get some background info going about the cell cycle. And feel free to enter any questions in the chat or unmute yourself to let me know if you have something that you want to tell me. So first, let's get started with the phases. So a regular cell generally has a couple of phases it goes through. So after division, we get interphase, which consists of G1, S, and G2. G1 is the first phase that a cell goes through after dividing, and it's where the cell focuses on growing in size, making energy, and making the proteins it needs, like mitochondria, ribosomes, and the ER, or the endoplasmic reticulum. Then we get to the S phase, where the main focus is replication of the DNA, where you go from one chromosome to two sister chromatids for a total of 46 chromosomes and 92 sister chromatids. Then we have G2, which is mostly a final stage of growth to make sure that the cell has enough resources to divide into two fully functioning daughter cells. And it's also a stage where quality control occurs, just making sure that everything is ready for that division stage. Finally, we get mitosis and cytokinesis, and that's really what we're going to focus on today. And that's where the cell divides all its chromosomes up into the daughter cells and the organelles and the cytoplasm as well. Now, a side note that I want to mention here is G0, and that's a stage that some cells can go into, like stem cells and nerve cells. And in this stage, what happens is cells go through their normal functions, like making proteins and energy, and they just don't progress further. And this can happen at any point in the cycle. For some cells, if the quality control checks are not passed, the cell can drop back into there before re-entering the cycle when it is ready. So also in this cycle, you notice there are some checkpoints. And these checkpoints are basically like the security checkpoint at an airport. Make sure that everything is good before moving on to the next stage so you don't have problems later. And these checkpoints can also be called restriction points. So the first checkpoint in the cell cycle you see is the one between G1 and S. And what we have there is the cell is just making sure that it's got the proper DNA from the parent cells. It's got the signals telling it to move on into the S phase. And it's got the machinery for replication, like DNA polymerase and so on. The next checkpoint that you get to in most cells is the one between the one after S phase and the G2 phase. And this one is just ensuring the proper duplication of the chromosomes and making sure that the cell is at a reasonable size and has enough organelles and cytoplasm to properly divide into two functioning cells. And both the first two checkpoints are generally regulated by a gene protein called P53. Now, the final checkpoint that you see here in most cells is the mitosis checkpoint. And this, which we'll get to later, is just a cell making sure that all the chromosomes are aligned properly for separation so that you won't have a dysfunctional daughter cell later. And this checkpoint can fail sometimes. So what controls the cell cycle? And to answer that question, we look at two things, cyclins and CDKs. So the cell cycle stage, or where this, which stage the cell is in, is regulated by levels of something called cyclins. And what these cyclins do is they, different cyclins change levels based on the stage that the cell is growing into. And these cyclins activate a protein called CDK. The activation of CDK causes a cyclin CDK complex, and that cyclin CDK complex phosphorylates some transcription factors. And transcription factors' jobs are to transcribe genes. And so the phosphorylation or the activation or the phosphorylation of specific transcription factors activates specific genes that are needed to progress in the cell cycle. And that's how the cell cycle is controlled by the signals within it. Now, dysregulation of the cell cycle can lead to something called cancer, which we've all heard about. And what is cancer? It's basically damaged cells that divide at an unregulated pace. And this kind of cell, or cancer cells, are controlled by two types of genes. Oncogenes, which when they're mutated or activated, promote cell division or promote unregulated cell division, and tumor suppressor genes. And mutations in these genes inactivate them and remove their ability to suppress or slow down the cell cycle and division. Now, a great example of these genes is TP53, which is a mutation of the gene making P53. And P53's activity is in DNA repair, and that's why it acts in so many checkpoints for the cell cycle. So when you mutate the gene, and that means you mutate the P53 protein, you don't get repair of the damaged DNA that the cell can find. And when you get division of a cell with damaged DNA, you end up with a cancerous cell because genes become dysfunctional, and those dysfunctional cells continue to survive and divide. Now, what a tumor is it's these cancer cells that undergo rapid cell division, or a group of cells that grow abnormally, which generally in the when related to cancer, we're talking about them growing super quickly and without regulation. There's also a concept called metastasis. And what that is some cancer cells that can spread from one area of the body where they're localized as a tumor through places like the blood or lymph vessels into another area of the body. And this can be caused by some cell factors like proteases and other factors that induce blood vessel formation near the tumor. So when these proteases lyse the cells surrounding blood vessels or lymph vessels, or these factors induce blood vessel growth near the tumor, some cancer cells may break off into the bloodstream or the lymph vessel and get carried somewhere else, where they then choose to stick somewhere and grow there as well. And that's what you can see in the top left picture over here. So I think that was a good bit of background. So I just want to go through a couple questions just to make sure we're getting the concepts properly. I'll give you guys about a minute for each question, and feel free to unmute yourself or put it in the chat if you think you have the answer. All right, I think that's been about a minute, and the majority of the answers that I am seeing is D, and that is correct. All of the others are potential phases for the cell cycle. S2 is not a phase in the cell cycle because cells don't undergo 2 rounds of replication before dividing. The first phase just corresponds to that kind of, I guess, sitting phase where the cell sits without progressing in the cell cycle and just continues to do its job in making proteins and energy. The S phase, corresponds to the synthesis of new DNA, and that happens when the DNA is replicated. We have G2, which is the second phase or the real quality control phase before the cell makes sure it has everything ready for replication or division. And we have cytokinesis, which is the actual physical separation of the parent cell into 2 daughter cells. And yes, feel free to let me know if you have any questions about that. But for now, let's move on to the second question. All right, so I think that's been about a minute as well, and it looks like everyone that answered the question got the right idea. I tried to make this one a little bit of a trick question, but it looks like you guys all got it. The answer is C, and that's because this checkpoint, or C is actually checked at the G2S checkpoint, where it's after the synthesis phase and it's checking to make sure that the cell was able to replicate or duplicate the DNA properly. The G1S checkpoint happens before the S phase, so it's just making sure everything from the parent cell was delivered properly and that it can perform synthesis properly. So I have the next question and the last one for this section here for you now. All right, so I'm getting the answer B from everyone, and that is 100% correct. Just to clarify or to explain one more time, because I know I didn't go over into too much depth earlier. An oncogene is one that when mutated speeds up the cell cycle by itself, and a tumor suppressor gene is one that when mutated so that it loses its function, allows the cell cycle to progress. So normally a tumor suppressor's gene function would serve to slow down the cell cycle or stop it even. And P53 is a tumor suppressor gene, because it is a checkpoint gene that serves to slow down the cell cycle and make sure that everything is correct before moving on. When mutated, it can't suppress the tumor formation or the cell cycle anymore. And so that's why it's considered a tumor suppressor gene. So there's really two types of cell division, and we're going to start with mitosis because it's the one that happens in pretty much every cell. And what mitosis is a process where the cell is basically splitting in two, and it creates two identical daughter cells and does not change the ploidy or the amount of genetic information in each of the daughter cells. Mitosis is really characterized by four main phases, along with cytokinesis, which is the physical splitting of the cell. And mitosis also happens in reproductive cells after their fusion or fertilization. So first we have prophase. And a couple things I want you guys to know because they're super important is the centrosomes. And a centrosome consists of basically a centriole, which is formed of 9 plus 2 or 9 plus 0 configuration of tubulin. Basically, you have 9 tubulin tubes in a circular configuration, and you have 2 in the middle, or you have 0 in the middle based on the bacteria or the animal cell. And what a centrosome is the centriole plus the free tubulin surrounding it. And it's basically a microtubule organizing center. So it's basically the foundation of any structure created during any structural building, I guess, that goes on during mitosis. What a centrosome does during mitosis is, especially in prophase, is it moves towards the opposite ends of the cell, as you can see in the pictures on the right. And it creates spindle fibers, which are bundles of microtubules. Microtubules are basically proteins that act as structure, and they're formed out of tubulin. And so these fibers, when they are attached to centrosomes, are called spindle fibers because the centrosomes connected during mitosis or meiosis are called the mitotic spindle. So from the centrosomes, we get two types of fibers, or I guess more than two. We can start with asters, which really anchor the centrioles to the cell membrane to make sure that the cell membrane isn't moving and that the centrosome can keep everything else in place relative to the cell membrane. We have kinetochore fibers and we have spindle fibers, which I will get to later. But the main part of prophase that if you're ever asked a question, the first thing that should come to mind is the nucleus or the nuclear membrane and the nucleolus dissolving or disappearing. So the membrane dissolves and the nucleolus, which is normally the site of ribosome production, also disappears. And the DNA that we have in this cell, which is normally in stringy, as you can see here, stringy spaghetti-like configuration, is called chromatin, condenses into distinct chromosomes that look like the X shape that we all know during prophase. So what's a chromosome? It is basically a set of genetic material that are not a set, but a piece of or a unit of genetic material that we inherit from our parents. And what's super important to remember is that it really looks like 1/2 of this X before it duplicates during the S phase. And that becomes super important for meiosis and mitosis. So I was talking about the mitotic spindle earlier, created by these two centrosomes and their attached fibers. So this is also known as the spindle apparatus, and it forms early between the centrosomes as they're moving apart. And you can see the fibers that are connected to both centrosomes here and some that are connected to the chromosomes as well. So The ones that are connected to each other are generally called the spindle fibers, and the ones that seek to connect to the chromosomes are called kinetochore fibers. What's a kinetochore, you ask? A kinetochore is just a very protein-dense structure that is extremely close to the centrosome on a chromosome. And the centrosome is basically an area that's tightly packed with DNA and doesn't really have any genes. And its point is, or its purpose is to hold the entire chromosome together. So the kinetochore proteins are generally tightly packed around that area as well. And that is where the kinetochore fibers bind during mitosis. So the next phase we have during mitosis is metaphase. And this is just establishing the centrosomes at opposite sides of the cell. So we have the astral fibers here. We have the kinetochore fibers connecting to the chromosomes. And we have other spindle fibers connecting these two centrosomes. So what I want to point out here is the chromosomes are now attached to the kinetochore fibers here, and all the chromosomes line up directly along this line called the metaphase plate. So this line is basically the center or equator of the cells, and all the chromosomes line up on the line. So half of the DNA from each chromosome or each sister chromatid will get one that goes to the top side of the cell and one that goes to the bottom side of the cell. So we'll have two kinetochore fibers attaching to each chromosome, one from each centrosome. And since the centrosomes and the mitotic spindle is anchored to the cell membrane, it can use the kinetochore fibers to realign the chromosomes by itself so that each chromosome is along the metaphase plate. And just a key thing I want to point out one more time is that in mitosis, the chromosomes are aligned along the midline of this plate. So here is where we have the spindle checkpoint, which I mentioned earlier, the M checkpoint. And this is where the cell is just making sure that all the chromosomes are aligned along the plate. So when we pull these chromosomes apart, half of the genetic material or a sister chromatid from each chromosome goes to one cell, and a sister chromatid of each chromosome goes to the other new cell. So the third phase we have in mitosis is anaphase. And a key thing to notice here is the chromatids and the chromosomes and sister chromatids are now breaking apart. How does that happen? Well, the centromere, the tightly packed area of DNA in the middle of the chromosome, disassociates. So the kinetochore proteins, their affinity for both sides of the chromosome decreases and breaks apart. So each sister chromatid is allowed to be pulled apart. And here you can clearly see the kinetochore fibers attaching to each sister chromatid, the astral fibers attaching to the cell membrane and anchoring the cell. And we also see other spindle fibers that connect both centromeres. So the motor proteins that are really moving the chromosomes and anchoring the cell membrane and moving them along the metaphase plate earlier in metaphase are kinesin and dynein. And that's just something I want to point out if it ever comes up in a paper. It might not. It's not necessary to know for cell division, but it may help you. So it's really dynein that is bringing the chromatids backward and closer to the centrosome on either side of the cell. And it will be kinesin that pushes. So you can think about the free ends of each fiber as the plus end and the end closest to the centromere as the minus end. So anything that goes towards the centromere is, the centrosome is called, or is done by dynein, and anything that goes towards the plus end is done by kinesin. So here we have the sister chromatids pulled in towards the centrosome by dynein. And that's what used to shorten these kinetochore fibers to pull them apart from the midline we had earlier, the metaphase plate. So then we also have, as I mentioned earlier, the use of kinesin, and that's done by elongating some of the microtubules, specifically the ones here in the center of the cells, some spindle fibers that connect to both centrosomes are elongated using kinesin, and basically they push the cell, which let's say used to be a circle, further and further apart, so it stretches further in this into this oval shape and further until it becomes easy to break it apart down the middle. And we'll see that in the next slide with telophase, which is some would say the final and some would say cytokinesis is the final phase. And this is where the mitotic spindle, where we had the centrosomes connected to each other and the cell membrane, it basically breaks back down into some free floating centrioles and free floating tubulin. So that's something that you should remember, but the real key part of telophase is the reformation of the nuclear membrane and the nucleoli. So what happens there is the sister chromatids or chromosomes that were pulled apart are now being put into their respective cell membranes, or not cell membranes, sorry, nuclear membranes, and nucleoli form as well to start making ribosomes again once the daughter cell is completely broken up. And in this nuclear membrane, the chromosomes get to uncondense back into chromatin and back to the form that they were in during interphase. And the key thing to remember here in mitosis is that both cells, if everything goes properly, will get an identical complement of DNA. They will be the exact same in terms of the genetic material that they have. So finally, we have the physical part of mitosis in terms of the entire cell. And that's just cytokinesis, where we basically make sure that both daughter cells have enough mitochondria, if you're an animal cell, mitochondria and ribosomes and other organelles, chloroplasts if you're a plant cell, and make sure that both daughter cells have enough and enough cytoplasm as well. So this physical process where the cells are basically torn apart or divided by a cell plate, as you can see in plant cells, can begin during anaphase or it can begin in telophase, and it ends shortly after telophase. So we do have a couple of differences in plant and animal cells, and in animal cells we have the cleavage furrow, which you can see as this little thin blue line here in the image. And what that is like, let's say a drawstring pouch or bag, where the string is basically formed by a band of another motor protein called actin. And what that does is it pulls inwards from the edges of the cell and from the entire band and pinches the cells apart. So it's like closing the drawstring bag. On the other hand, in plant cells, we don't necessarily have that just because plant cells have a cell wall. So what forms in the cell is a cell plate, and that cell plate is made by vesicles that come from the Golgi body just before mitosis begins. And those vesicles contain more cell membrane and also a cellular matrix that later forms the cell wall. So these vesicles all come together in the middle of the cell to create the new cell wall, just because the cell wall is too stiff in the original cell to pull it apart like we did for the animal cells. Again, a key thing I want to mention here is after mitosis, the daughter cells do retain their 2N ploidy. So they still have the complement from their mother and the father of the plant or animal cells or bacterial cells. And what really happens to these cells after post-division is they're basically programmed to die after 20 to 50 divisions based on the type of cell, just because the older the cell is, the more likely there are to be deleterious mutations that could really turn them into cancerous cells. So that was pretty much mitosis in a nutshell. I do have another set of questions for you guys though. And as always, I'll give you guys a minute or less if you guys seem to know it really quickly. Feel free to ask any questions or type them in the chat. All right, perfect. So I am getting a lot of E's, and that is correct. It is in telophase that the nuclear membrane starts to reform along with the nucleoli and the chromosomes uncondensing back to chromatin. Here's another question, just to make sure we get. All right, so this was a concept I didn't really touch on too much just because I wanted to come back and explain it later with this question. So P53 is a protein that regulates DNA damage and repair. So we know it's not a motor protein or a structural protein. And from here, we'll work our way down the list. So A, actin, C, kinesin, and D, dynein, I hope I'm saying it right, are all motor proteins, but they're used in different purposes. So for the purposes of mitosis, actin is the protein that actually creates the contractile band in animal cells. It's the one that does the drawstring pouch thing where it basically pinches the drawstring shut and the two cells apart. Kinesin and dynein are mostly used in with respect to mitosis on the microtubules. And what kinesin does is it moves things to the plus end. It basically grows the microtubules or lengthens them. And dynein is the opposite. It goes the opposite way on the microtubules. It goes towards the centrosome, and in other words, it can shorten the microtubules. So E-tubulin is the structural, I guess, unit of microtubules. So a microtubule is formed by many segments of tubulin. Yes, I can definitely go back. And so the contractile band is basically one cell that you can imagine like a circle and like here. And as it gets pushed apart, you can see it turning more into this oval-like shape. And finally, we get to the point where there's just a lot of actin in this central area here, and it uses the cell membrane as an anchor point to basically pull the cells apart or pinch them apart. Does that make a little more sense? Yes, actin it's what forms the contractile band that's part of the cleavage furrow. Not sure what you mean, Brian. Actin is like it says here, it's a ring that basically tightens and closes up. And the closing up of that ring is what creates the cleavage furrow when it does the pinching action here. Does that
Oh,
make a little more sense?
so this is not the line that's connected to the kinetochore, which will responsible for pulling the chromosome onto
No,
them.
no.
That's not a line.
Yeah,
The,
that's a different. These lines here are formed by microtubules, which are made up of tubulin subunits. And that is related to the centrosome and the chromosomes and anchoring the cell. The cytokinesis or the cleavage furrow is done purely by actin because in telophase, the entire mitotic spindle breaks down. So you don't have any more microtubules or tubulin that is holding the cell together. Does that make a little bit more sense?
yes, thank you so much. I was a little bit
Yeah,
confused on the terminology.
no worries. I completely understand. There's a lot of different terms and a lot of the words sound similar as well. And here is another question just to clear up metaphase and prophase. So this is a question I just wanted to use to point out part of mitosis, just because it is very different when you consider meiosis, specifically meiosis 1. There are 2 spindle fibers that attach to each chromosome during mitosis, and one spindle fiber from one centrosome and another spindle fiber from the other centrosome on the other side of the cell. And you can see that here in metaphase, where let's use this chromosome as an example, has one spindle fiber or kinetochore fiber coming from the bottom centrosome here, and another spindle fiber coming from, or a kinetochore fiber coming from the top centrosome here. And that is because those 2 spindle fibers will break the chromosome apart when they both pull on the chromosome. And I believe I have one more question for you guys about mitosis. All right, so this one was just to make sure you guys understood the different types of fibers that exist during mitosis, and there are a lot. So I completely understand. So we'll go through them one by one. Kinetochore fibers are the fibers that go from the centrosome to the chromosomes. So the ones that are attached to each chromosome right here, they can be called kinetochore fibers because they bind to the kinetochores on each chromosome. All right, and then the next one is astrofibers. Astrofibers, to go back and use an example, are the ones here that you see between the centrosome and the cell membrane closest to them. And what they do is they basically attach the centrosome to the cell membrane to anchor the cell when they perform things like realignment of the chromosomes. And then we have centriole fibers. That's, I guess, a phrase that I just made up. And the last thing is mitotic spindle fibers. So those are the fibers here we see in anaphase that don't connect to the chromosome because those are kinetochore fibers, and don't connect to the cell membrane because those are astrofibers, but instead connect the two opposite centrosomes. And those are mitotic spindle fibers. And when they're connected to each other, they grow at the ends that connect. And so they push each other apart. And so the centrosomes apart and the cell apart as well, because each centrosome is anchored to the cell membrane. I hope that makes a little more sense, and definitely let me know if you'd like me to explain it a little bit further. But if not, I have one last question on mitosis. I will say what the correct answer is, I guess, when right before I explain this one, just to let you know. All right, perfect. So I was asked to explain the previous question, or the right answer to that one, and that is D. They're just mitotic spindle fibers that attach to the opposite centrosome and grow, pushing each centrosome apart and the cell apart as well. So D is the right answer for this one. Yes, no worries. So for this question, C, prophase, is the right answer, because in prophase, the nuclear membrane disappears and the chromatin has condensed into the chromosomes, and that way it can later align up along the metaphase plate during metaphase. So that's basically all I have for you guys on mitosis. So we have meiosis, which happens only in sex cells, also known as gametocytes. And it tends to create four non-identical daughter cells, also known as gametes, because they're sex cells. And you can see that here, all the way to the right. So the first thing with meiosis is I want to compare it to mitosis, because there definitely are some similarities, but there are also some differences. So the similarities are that we have replication of the DNA before the cells or cell starts dividing. We also have the genetic material, or the chromosomes, separating into the daughter cells. And we also have the mitotic spindle that sets up and divides cells in both mitosis and meiosis. So now we get to the differences. So there is a difference in mitosis and meiosis because there are different numbers of division rounds, I guess. Mitosis goes through the single round of division, where you go from one parent cell to two daughter cells. And meiosis has two rounds of division, where it divides into two daughter cells, and those two daughter cells divide again into another set of two daughter cells. So we have reductional division for meiosis 1. And what that means is we get the ploidy, or the 2n state of the cell, down to n, where you only have one copy of each chromosome. And what you can see that here from meiosis, where you have the homologs, where the cell is diploid, and you have these separated into the haploid type, where you have only one copy of each gene. And that's called reductional division, because we are reducing the variety in genetic material. So we go from 2n here to n for each of these cells here. The meiosis II, however, is very, very similar to mitosis in that it's equational division. The ploidy, or the amount of genetic material in the cell, does not change. For mitosis, you go from 2n to 2n. And for meiosis II, you go from n to n. Now, just to let you guys, or I guess just to tell you guys, there are 23 chromosomes that we start with, right? 23 sets of chromosomes. And after replication, there are sister chromatids, which is basically the same genetic material in two different physical components. And so they're identical strands of DNA, just connected by a centromere. When we have the S phase in meiotic cells, we get 23 homologous pairs. So we have 23 chromosomes from our dad and 23 chromosomes from our mom, and they have the same genes, although the allele for the gene may be different. So we have 23 homologous pairs, which means 46 chromosomes. And once those chromosomes have replicated, we get 92 sister chromatids. And just to explain a little further, homologous pairs can also be called bivalents, and it just means a paired maternal and paternal copy of the same chromosome. So for meiosis, we can look at this bottom largest chromosome here, and we can say the red is the mom. So we have two sister chromatids and one chromosome from the mom. And we have the blue is the dad, where there's two sister chromatids, but one chromosome for the dad. So I mentioned earlier that meiosis I is the different part of meiosis. So we'll cover that. So it's still a little similar in that we still get the chromatin condensing into distinct chromosomes, and the nuclear membrane, as you can see here, and the nucleoli all disappear or dissolve. We still get the formation of the mitotic spindle or spindle apparatus, and the kinetochores that later connect to the kinetochore fibers. However, we have what is called synapsis, where the homologous chromosomes from our mom and our dad can line up by themselves before they're connected to the kinetochore fibers. And so the homologs, or the homologous chromosomes, and the four sister chromatids can create something called a tetrad, which you can see here, where they're aligned with each other. And they can be held together by a complex of proteins called the synaptonemal complex. So when they're held together, since the genes are so similar and everything is so similar on the chromosomes, there is a chance of something happening called crossing over or recombination. And that's just an exchange of the DNA on each of the homologs with the other. And you can get this once, like we see here in the bottom right picture, on the two chromosomes, or you can get it two or three different instances in different places of the chromosomes. So here we see it on the bottom leg of the chromosome, and we can see it on the top leg as well. And this doesn't happen in mitosis. But this crossing over or recombination allows genes to be transmitted separately. So it allows diversity, since we're not getting the same chromosome transmitted between generations, but we have this chance to mix things up a little. And the point of contact between the two homologous chromosomes, as we see here, is called a chiasma or chiasmata, singular and plural. And this recombination really just exemplifies Mendel's second law, or the law of independent assortment, which says that the inheritance of one gene doesn't affect the inheritance of a second gene. And we see that because if the capital B gene came from the dad, and it's on the same chromosome as a capital C gene, they don't necessarily get inherited together because of things like recombination, because we now see the capital B gene is transmitted with the lowercase c gene. So in terms of the actual application of meiosis I, this is where going to try to say this word, oocytes or oocytes are, or egg cells, things that develop into egg cells, are stopped in division. So when a female is born, the egg cells or primary oocytes stop here at prophase 1 until they start to have their menstrual cycle. So at the first menstrual cycle, they will they will then start the cycle again and go from prophase 1 to the end of meiosis 1 every single time they have the menstrual cycle. And this is also where primary spermatocytes are. This is the stage where primary spermatocytes are before they progress into secondary spermatocytes. So then we have metaphase 1, which compared to prophase is pretty simple. It's a lot like mitosis, but there's, again, are a few key differences. So the main thing here is we still have the spindle fiber or kinetochore fiber attachment to the chromosomes. However, and you'll notice this in the picture, since we have a tetrad and the chromosomes don't line up in a line like we used to see, we see them now line up in pairs. We only see one kinetochore fiber attached to each homolog. So instead of two kinetochore fibers from from one from each centrosome attaching to each chromosome, we have one kinetochore fiber that attaches to one homolog. So in this case, they in meiosis 1, they all line up alongside the metaphase plate. They don't line up on top or splitting along the line because we get the separation of the homologs into two different cells. So what the imaginary line in this case would pass right through the center of each of these chromosomes. And this happens because we have paired homologs here, and you don't get that in mitosis. So we have primary oocytes in prophase 1. Secondary oocytes are stopped here in metaphase 1. Next, we have anaphase 1, which again is slightly different because we have homologous pairs. What happens here is we get the separation of these homologous pairs. So we may see the paternal copy of one chromosome go to one side along with the maternal copy of another chromosome. So the same genes but different alleles are being separated into different cells, if that makes sense. And feel free to ask me if it doesn't. So what this is called is disjunction, where you get the separation of chromosomes and them being pulled into a different area or pole of this cell. And this shows the law of segregation. Because the homologs separate randomly, you don't get all the paternal chromosomes in one daughter cell and all the maternal chromosomes in the other daughter cells. They're all random. So the alleles also separate or segregate randomly. And that explains the law of segregation, which says that not all alleles are transmitted together. So you won't inherit the exact same set that your dad had. So again, we have the motor protein movement, the mitotic spindle fibers pushing each other apart, and so the cell apart. We have the kinetochore shortening, which pulls the homologous chromosomes inward. And we finally have non-disjunction, which is a case where meiosis doesn't work properly and these homologous chromosomes don't separate properly. So in this case, they are separating properly. But if they don't, you could get both of these homologous chromosomes going to one side of the cell and the other daughter cell doesn't get any chromosomes. And the end product of non-disjunction is that one or more gametes have too many or too few copies of the chromosomes. And as you can imagine, that's super destructive for the cell or the offspring. Yeah, for sure. So yeah, non-disjunction is super destructive for the cellular offspring because you have too much gene expression, or you don't even have the gene to express. And that's why things like trisomy 21 or Down syndrome is super destructive for the person that has it, unfortunately. You can also see this in sex chromosomes, where you get something called Klinefelter's syndrome, where you have 2 X's and 1 Y, so 3 sex chromosomes, because they didn't go through anaphase or disjunction properly. And you can also have Turner syndrome, where you only have 1 X chromosome and you don't even have another X or a Y. Again, that's because of non-disjunction, where you ended up being the cell that got that didn't get the proper amount of sex chromosomes. So here, the microtubule elongation would be on fibers like this one in the dead center of the cell, and these two on the top and bottom ends of the cell, where they would basically grow and push against each other as they grew. And that push, since they were joined, would push back on each of the centrosomes and push them, this one further left and this one further right. And since each centrosome is anchored to the cell membrane, it would also push the cell membrane along with them and naturally create this tightening in the middle. No, we don't need to know each specific type of non-disjunction disease. I'm just letting you know because these are 3 of the most important ones that came up for me several times. I believe I do have a question about it later, but the one I would 100% recommend you understand at least a little is trisomy 21 or Down syndrome, where you have 3 chromosomes for the 21st pair of chromosomes, because non-disjunction occurred. So then we have telophase 1, which is, or telophase 1 cytokinesis and interkinesis, because meiosis goes through 2 rounds of division. And this is, again, super similar to mitosis because you see the mitotic spindle breakdown and you see the reformation of the nuclear membrane and nucleoli. But the key difference from mitosis is, excuse me, you get haploid daughter cells because each cell has 1 copy of the 23 chromosomes. You may have gotten your mother's pair for one and your father's for the other, but you only have 1 copy of each chromosome, and that's super important in meiosis. That also makes you haploid because you have, or N, because you have 1 copy. Again, cytokinesis is the separation of the cellular components and making sure that both daughter cells will have the materials necessary to survive on their own after. But since meiosis goes through 2 divisions, we have something called interkinesis, and this is the gap between divisions that may or may not occur. And in this phase, if it happens, we get a partial uncoiling of chromosomes back into that stringy spaghetti-like chromatin state. Now, this phase, or after meiosis 1, is where we get secondary oocytes. And what happens in meiosis 1 is the primary oocyte gets divided into a secondary oocyte that gets a large share of the cytoplasm from the parent cell, and a polar body, which is basically fragments of a daughter cell that have very little organelles or very little cytoplasm. And that happens as a result of unequal cytokinesis. And this is also the stage where secondary spermatocytes are created. So then we have meiosis 2, which is super similar to mitosis, so I'm not going to go into too much depth here. It's similar to mitosis because it's equational division. We're not losing or splitting apart the genetic material any further, because all we're doing is separating the sister chromatids. And since the sister chromatids have the exact same genetic material, we aren't losing any when we split them apart into separate daughter cells. So it goes from N to N. So the 23 chromosomes and 46 sister chromatids at the beginning of meiosis 2 goes to 23 chromosomes and 23 chromatids at the end of meiosis II. So just to go over it again, since it's so similar to mitosis, in prophase, the most important part is that the nucleus disappears, the chromatin condenses, and the spindle apparatus forms that to anchor the cell and to line up and separate the chromosomes or chromatids. In metaphase 2, we get the chromosomes attaching to the kinetochore fibers, and the kinetochore fibers will then realign them along the metaphase plate. In anaphase 2, we get the centromeres dissolving and the kinetochore fibers shortening by the use of dynein to separate the chromosomes. And in telophase 2, we get the reformation of the nuclear membrane. And we also get the uncondensation of chromosomes back into chromatin again. And also important, the mitotic spindle breaks down here to allow the cells to split properly in cytokinesis, where the actin band, again, in animal cells is drawn and pinched tightly to separate the cells, and the plant cells create a cell plate to pinch it apart. And this meiosis II is triggered for the egg when the sperm fertilizes it. So only when you get fertilization does the egg actually undergo meiosis II to create the actual daughter cell that the sperm will enter. This is triggered by something known as the acrosome, which is a group of enzymes on the sperm's head. When it penetrates the egg or the oocyte, it triggers a reaction that causes meiosis II, and you generate an ovum or egg, and that's where the sperm actually penetrates. And that's what you see here, where you get meiosis II creates spermatids that then mature into sperm, and then it creates ovum, which the sperm then actually fertilizes. The thing with meiosis II is in eggs, you can have up to 4 daughter cells because of the creation of polar bodies, which aren't viable as cells to grow and complete their own division, just because they're left with so little of the organelles and cytoplasm. So on the other hand, we get the ovum, which is just a large cell with tons and tons of RNA, organelles, and cytoplasm to immediately start division and growth once the sperm fertilizes. What you see here in the sperm is you see the head, which is this little ovalish piece that contains the acrosomes at the tip to penetrate the oocyte, and the enzymes within that acrosome, along with the nucleus, which contains a haploid set of the father's genes. We have the mid piece, this thick portion of the tail here, that, or this thick portion before the tail here that contains tons of mitochondria to help the sperm move. And we actually have the tail, which spins to create motility and move. So the formation of the zygote is when the sperm enters the egg and the nucleus releases the genetic material or DNA into the egg, and it turns the zygote back into a diploid. As you can see here, this is from the mother and this is the DNA from the father. And what you want to know about the zygote is the ovum or the egg basically contributes everything here except for that half set or haploid set of chromosomes that the sperm donates. So when the sperm nucleus or pronuclei fuses with the ovum, that's when you get the actual zygote. So I have some questions for you guys about meiosis, and we're almost done. So please hang on for not too long, hopefully. Perfect. I'll try to go a little bit faster so we can get you guys out of here without taking too long. Yes, it is B that causes Down syndrome, and that's 3 copies of the 21st chromosome. You may get 2 from your dad and 1 from your mom, or you may get 2 from your mom and 1 from your dad. A, the XXY creates Klinefelter syndrome, and C, X naught is Turner's syndrome, where you only have 1 sex chromosome from your mom. All right, the next question. All right, perfect. So in meiosis 1, the chromosomes do line up alongside, and that's because there's 2 homologous chromosomes, and each one lines up alongside the metaphase plate. So one homolog will go to one daughter cell, and the other homolog will go to the other daughter cell. I wanted to point this out because this is a key difference from mitosis. Perfect. Yes. If an egg is left unfertilized, it will be left as a secondary oocyte, and that means it has gone through just meiosis 1. And meiosis 1 results in 1 daughter cell that is the secondary oocyte and 1 polar body. So you will get 2 daughter cells from the primary oocytes, but only 1 is viable to continue to divide in becoming an egg. Here you go. Here's the next question. All right, so at the end of meiosis 1, since it is reductional division, sorry for the extra comma there, I just noticed, we go from 2N, which is a complement from the father cell or the father and the mother, to to N, which is a complement, only a single complement of the 23 chromosomes from a mixture of your father's and your mother's genome. 2N to 2N occurs only in mitosis, and meiosis 1 goes from 2N to N, since you're splitting the homologous chromosomes and you only end up with 1 copy. And meiosis 2 goes from N to N, because you're splitting sister chromatids you're splitting chromosomes which are sister chromatids, so they have the same genetic material. All right, guys, we're almost out here. Here's the next question. All right, perfect. So prophase 1 does prove the second law of Mendel, where he said that genes do not have to be inherited together. And you're right in anaphase 1, proving the law of segregation in saying that alleles aren't necessarily inherited together because the homologs separate, taking its copy of its allele with it into two different daughter cells. All right, I have one more question for you guys. All right, so at the end of meiosis 2, we will have an N ploidy, so 23 chromosomes. And since during meiosis 2, we split up the sister chromatids into two separate daughter cells, we won't have sister chromatids in any of the gametes since they are being split up into separate daughter cells. So the correct answer is B. Yes, perfect. A is at the end of meiosis 1, since we have an N ploidy, but we also have a sister chromatids in the daughter cell. Perfect. So before we leave, I just want to remind you guys that we are a nonprofit and any donation would greatly help us. And don't forget to spread the word about us because we'd love to reach out to as many people as possible. So I'll leave this up on the screen for a couple seconds. And the last slide I have today for you guys is thank you. If you have any questions or you'd like to send us feedback, feel free to email me at the address you see here, or feel free to email MCAT101 with the questions you have here. And again, if you'd like to be added to the newsletter, send me your email and I'll make sure you're added. And, oh, sorry. Please fill out the survey that we will be sending to you guys in an email. I'll be sticking around for a couple more minutes. I'll stop recording. If you guys